Gentlemen, my name is Kamala Shireen Lakdir, and I'm the U.S. Ambassador to Malaysia. Thank you for tuning in this Saturday morning for a very timely and interesting public health talk featuring distinguished scientists, Dr. Adeeba Kamarulzaman from the University of Malaya and Dr. Frederick Altice from Yale University in Connecticut, the United States. This is the second public health talk we have held with Dr. Adeeba and Dr. Altice, with the first occurring last May. A lot has happened then, and today we will revisit the joint research between the University of Malaya and Yale University on COVID-19 treatments and hear more on the progress that they've made together. We've also heard very encouraging news over the past few weeks on vaccine effectiveness from Pfizer and Moderna. I look forward to hearing from our experts on how they interpret that news and what it will mean for fighting this pandemic going forward. As a non-scientist, I am in awe of the critical work being done by researchers and medical professionals around the globe. The research into COVID vaccines and treatments may also prove to be useful in many other applications as well. I'm interested in hearing the professor's views on the potential implications of medical advancements from COVID-19 research and where this could lead us together. At this unprecedented time, the United States is leveraging all resources available to help ensure that safe, effective, affordable, and widely available vaccines, therapeutics, diagnostics, and other medical products are developed, manufactured, and distributed as quickly as possible at the scale needed to rapidly reduce the human, social, and economic toll of the devastating COVID-19 pandemic. In the United States, we have been at the forefront of the global COVID response with our vaccine development program, having already awarded over $12 billion and with $6 billion more planned. While it may be too early to tell for sure, the most recent news from clinical trials seems to suggest that we may meet our target of making the initial doses available by January 2021. U.S. investments in COVID-19 innovation will also benefit the global community. In addition, the U.S. government has specifically allocated more than $20 billion to the international response. Here in Malaysia, the U.S. government has been funding critical health work for many years. For example, research on emerging zoonotic diseases such as COVID-19 and Nipah virus through USAID's PREDICT program, which is doing work both in Sabah and in the peninsula, and with funding for the National Institutes of Health. I just want to stop here because part of our NIH work is through Dr. Fauci. So I'm always proud to see him on, knowing that he is also contributing to our collaborative work in Malaysia. We also support the Malaysia One Health University Network, which works to strengthen the capacities of universities in Malaysia and the region to enhance global biosecurity. During this terribly difficult year, we have also been able to support people in need in Sabah with food donations and an ambulance in collaboration with the International Red Cross and Red Crescent. In July, we helped enhance the COVID-19 testing capacity of the Public Health Laboratory in KK with $300,000 in additional supplies. But also I'd like to highlight that the US private sector here in Malaysia, through My AmCham Cares, has donated over 7 million ringgit to COVID-19 response and relief efforts. And they continue to make a call to all their companies to support Sabah at this critical time. Now it's my great pleasure for me to welcome our moderator, who will then introduce our expert speakers this morning. This section will be moderated by Dr. Sean Thum, a medical officer who has served as a COVID-19 frontliner in Johor. Dr. Sean is also an alumnus of the US government's Young Southeast Asian Leaders Initiative, YSEALI, which aims to strengthen leadership development and networking in Southeast Asia. Thank you, Dr. Sean. So I'm really looking forward for this engaging and interesting discussion. I just would like to add one thing more at the end. So Dr. Adeeba and Dr. Altice represent for me the most critical and important people-to-people work that we have between the United States and Malaysia. I always think of our cooperation on public health as a total blessing for both our countries. I always think of it as doing good for everybody. And I knew about their work together before I even became an ambassador. So I'm very proud to introduce them today and for us all to hear of their incredible work together. Thank you very much. And Dr. Sean, over to you.
Thank you, Your Excellency, for the opening remarks. I'm sure everyone in attendance, including myself, is as excited as yourself in anticipation of our program today. Hello, everyone. My name is Sean Thum, a YSEALI alumnus and doctor managing COVID-19 patients in Sabah. The mountain you see in the background is Mount Kinabalu, the pride of Sabah. I'm extremely delighted to be in the presence of two world-renowned experts, Prof Adeeba of the University of Malaya and Prof Altice of Yale University. For your information, this is the second time we are meeting our speakers to discuss about COVID-19. In our first session last May, we spoke about the race to COVID's end. It is 6 months to the date since our first session. Where do we stand right now? Well, we shall find out in a bit. Before we begin the session, I would like to invite all of you who are joining us via Facebook to fully utilize the comments section, submit your burning questions, and together learn more about this topic. A brief introduction of our speakers. We have Professor Adeeba Kamarulzaman, Dean of the Faculty of Medicine and Professor of Medicine and Infectious Diseases at the University of Malaya, as well as the President of the International AIDS Society. Joining Professor Adeeba on the panel is Professor Frederick, Altice, Professor of Medicine, Epidemiology and Public Health at Yale University School of Medicine and School of Public Health. Without further ado, let us hear from our speakers. Prof Adeeba, over to you.
Thank you very much, Sean, and good morning, everyone, and greetings to Ambassador Lakdir and my good friend and colleague, Professor Rick Altice from Yale. And it's a real pleasure to be back 6 months since our first forum together. And boy, have things moved in the world of COVID science. And, in the 15 minutes that we have this morning for me to summarize some of the findings, I hope I can do it justice to as Ambassador Lakdir mentioned, the incredible amount of work that's going on around the world collaboratively to advance COVID, not just treatment and vaccine, but also public health interventions. So actually, before we move on to the excitement of the vaccines, I'm actually going to spend a little bit of time summarizing some important key findings on viral transmission as well as treatment, because I think although the vaccine has given us all a relief in terms of the future of the pandemic, but realistically, we will probably not see its impact, at least locally, for a good at least 12 months or so, because as is now famously said, vaccines don't work, it's the vaccination that works. So the challenge of scaling up vaccination across the country and around the world is not small. So we will still need to live with the virus and stay on the public health interventions that have proven to work. So with that, if I could have the slides, please. Okay, and the next slide. So I think in the beginning of the pandemic, the concern was, of course, and rightly so, that the virus was mostly spread by droplets and also through fomites or the environment, the surfaces. But in recent months, attention has also been given to the potential spread of the virus, potential airborne spread of the virus, which can implicate the transmission even further. I think that's an important new finding that has relevance in how we continue to try and mitigate the pandemic through public health interventions, so non-pharmaceutical interventions. The other major finding in the last few months is also the importance of ventilation and air circulation. It is my honest opinion that I think not enough attention has been given to this important, somewhat inexpensive in many ways, simple intervention like opening doors and windows in enclosed settings that can contribute to reducing the potential risk of transmission, particularly indoors. Next slide. And I think if you all recall, back in January, we were all washing our groceries and keeping meals in plastic bags and leaving them outdoors for 2 days for fear of the virus being on our mail and boxes. However, I think recent scientific findings have shown that the viruses don't linger on these fomites for as long as we used to think, and that surface transmission plays lesser importance than that of airborne and airborne transmission. So masking and physical distancing and having good ventilated areas now should be the focus of our interventions until we get the vaccines. It's not to say that we shouldn't be washing our hands, that remains important, but the obsession with those massive cleaning, it has become less important. Next slide. Next slide, Hafizul. Okay. The other, in terms of public health understanding of virus transmission, is that a small number of people can generate large clusters, and the importance of the super spreading events, as is mentioned here. In a study in Japan, they contact trace 110 people, and in 10 indoor clusters, and as it turns out, 27 primary cases generated nearly a quarter of secondary cases. And this has been seen in many other clusters around the world, and including Malaysia. As you all will recall, our first large cluster was in a religious gathering, the tabligh gathering, that lasted for at least a good 3 months, and created and resulted in many people becoming infected. So I think in terms of public health interventions, the messaging around not having large gatherings must remain, particularly in indoor settings like gyms and restaurants, and as you can see here, choir practices, because that singing action will spread the virus. Next slide. This study, this very interesting study of a super spreading event in Japan, again, aims to show the importance of ventilation. As you can see from this cartoon, those who were seated around patient 0 at table A were those who became infected, whereas even though the distance between those who were sitting at table D and, if I can call it, table E, wasn't that much different from those who were sitting at C and B, but because this air circulation is as such, as you can see in that diagram, many who were seated in that route of that air circulation became infected. However, those who were seated away from that air circulation were not. And once again, this aims to show the importance of aerosol transmission, as well as the importance of good ventilation and air circulation, which, you know, I think we need to pay more attention to. Next slide. So in summary, in terms of the SARS-CoV-2 transmission, crowded enclosed spaces facilitate SARS-CoV-2 transmission. And I think Rick and colleagues from the US, so all of you are about to celebrate Thanksgiving, and I'm reading in all the US newspapers calling for, unfortunately, that you shouldn't be having celebrations as you normally do in your homes, and to kind of minimize the family interactions and the size of the family interactions, which is unfortunate, but you know, I think there's enough evidence to show that indoor settings which are poorly ventilated, and as it would be in the US now, where you're facing winter, is an important route and source of transmission. And of course, the longer duration that you're together, the greater the risk of transmission. Next slide. Okay, moving on to advances in treatment, and here's a summary that was shared with me by Dr. Suresh. As you know, Dr. Suresh leads Malaysia's clinical COVID response, and just a reminder that for the most part, the at least in Peninsular Malaysia, the large proportion of people who have been diagnosed with COVID-19 have either mild illness or asymptomatic illness. In this slide that was shared with me by Dr. Suresh, in Tawau, in Sabah, that you heard from Ambassador Lagdir, of how large the outbreak there is, the situation is slightly different. There's a much more there are a lot more people who present with moderate or severe illness in Tawau, and that, I think, has to do more with the geographic situation in Sabah, where people live a lot further and have difficulty accessing the medical facilities, and perhaps also in terms of knowledge about the disease. But here in Peninsular Malaysia, the breakdown of disease severity pretty much mimics that of previous reports from around the world, where for the most part 70% or so of people are either asymptomatic or have very mild disease. Next slide. And the good news is, of course, in the last 4 or 5 months or so, with intense research going on around the world to look for treatment, we have treatment that has been successful in reducing mortality all around the world. And also that treatment has been based on better scientific understanding of the natural not just the natural history of the disease, but also of the immunopathogenesis of the disease. I'm using big words here, but in terms of the early insult from the direct viral causes, causing viral pneumonia, followed by the second week where you see deterioration and complications largely due to the host inflammatory response, as you can see from the orange triangle here. So by understanding what causes the deterioration and complications to the disease, we have been able to design better treatment modalities. And as you know, worldwide now, the recommendation is to treat those who deteriorate or have severe illness with corticosteroids, dexamethasone, based on a very large study from the UK. Next slide. However, there are many ongoing studies, and as you can see here, from different therapeutic classes of drugs, either those which have antiviral properties or immunomodulators. Next slide. Just to summarize the studies from one of the antivirals, Remdesivir, which has yesterday been recommended by the WHO as not suitable for treatment of severe COVID-19. This is a meta-analysis of randomized controlled trials involving Remdesivir. And as you can see, data from more than 7,000, this meta-analysis accumulated data from over 7,000 patients across many trials, which included the Solidarity trial that was convened by WHO, which Malaysia participated in. And what this meta-analysis showed, that there was no important effect on mortality, as well as need for mechanical ventilation and time to clinical improvement in those in the Remdesivir arm. And therefore, as of yesterday, the WHO no longer recommends Remdesivir for treatment of severe COVID-19 infection. Next slide. If you recall, back in October, the FDA approved Remdesivir for treatment. Next slide. As mentioned, following this large meta-analysis, it's no longer recommended. So next slide. Moving on to corticosteroids, which currently remains the drug of choice for treatment of severe infection. The announcement of the halting of this clinical trial back in August in the recovery trial conducted by Oxford and a whole consortium of research centers in the UK. It was a very large study that looked at the effects of Kaletra, lopinavir, ritonavir, dexamethasone, hydroxychloroquine, or azithromycin, and had more than 11,500 patients enrolled in 175 hospitals around the UK. The study, as you recall, was halted prematurely because of the remarkable benefit shown in patients receiving dexamethasone arm of the study. So next slide. So what, this is just one result from that study, and in terms of mortality, the primary outcome showed that those receiving dexamethasone compared to usual care had a lower mortality among subsets receiving invasive mechanical ventilation or oxygen alone. So to this day, dexamethasone is the recommended treatment for patients requiring oxygen or have progressed to needing mechanical ventilation. Next slide. again, a meta-analysis of studies involving corticosteroids once again shows that corticosteroids show significant benefit in patients requiring oxygen or mechanical ventilation. Next slide. So it's recommended by most international, all international guidelines as the drug of choice. Next slide.
Hello, Prof. Sorry to cut in. We do have just a little bit of time left for you. Sorry.
Yes, very quickly, wrapping up in terms of another class of drugs, the immunomodulators, either the soluble IL-6 receptor, the anti-IL-6 receptor modulators have also been used in many clinical trials, including the one that we are performing here in Malaysia, the STORM study, based on the understanding that the deterioration is related to the inflammatory storm or the cytokine storm. However, studies using, next slide, Tocilizumab have been quite mixed. In some studies from Italy and others have shown no improvement in patients receiving Tocilizumab. Next slide. A study from Yale, which Rick may be able to, I'm not showing the slide here, but in an observational study from Yale has, however, shown an effect of Tocilizumab, a positive effect of Tocilizumab in patients receiving mechanical ventilation. So with the mixed reports from various studies with Tocilizumab, current guidelines state that it should only be used in the setting of clinical trials. Now, along with the advice from WHO not to use Remdesivir, next slide, out came the excitement, next slide. This release, again from Oxford, of a study called the REMAP-CAP trial. Although the details of this study have not been shared, but in this press release from Imperial yesterday, it shows that treatment with Tocilizumab has reached a key efficacy endpoint among critically ill patients with severe COVID-19, which shows that they were more likely to improve in patients receiving Tocilizumab versus those who were not. So I think there is much to be learned still from the role of the immunomodulators in the treatment of those who are developing complications from cytokine storm or those who already have severe disease. Next slide. And keep going, so I'll just, in the interest of time, just give a very quick update of our very, next slide, own STORM study, next slide. Next. Okay. Back in March, when Rick and I were discussing how best to manage patients with COVID-19 who were at high risk of progression. Remember, back in March, we were trying all kinds of treatment, including interferon, including Kaletra, including hydroxychloroquine. But our own personal, or at least Rick's and the Yale team's personal experience with Tocilizumab was very favorable. And so we decided to design a study to compare Tocilizumab versus corticosteroids. Again, remember back in March, steroids were in fact not recommended based on experience with SARS and MERS. But we decided that based on the understanding of the disease, we were going to do this head-to-head comparison with Tocilizumab and steroids. So following many intense discussions, we designed this study, and which received approval, ethics approval, both at UMMC as well as Ministry of Health hospitals, because this is a collaborative effort with Dr. Suresh and team at Sungai Buloh and a few other Ministry of Health hospitals. And then we had the pleasant problem of the pandemic kind of in Malaysia really going away, with us not being able to enroll any patients because of the good public health interventions that we had and very, very low levels of infection. And so for many, many months, we were not able to enroll anyone, even after receiving the ethics approval. However, unfortunately, since September, we've had a surge once again, and so we now have 18 patients enrolled in this trial. We are aiming for 300 patients, 150 in each arm. It's too soon for us to be able to share with you any results of that study, but we've also modified the protocol, taking into account new knowledge around corticosteroids, dexamethasone, from studies that have been happening worldwide. So with that, I just want to say that, as has been alluded to by Ambassador Lagdir, these kinds of studies and the immense knowledge that have come about from research, not just basic science research on the virus, public health, treatment, vaccine research all over the world, have only been possible because of collaboration. And again, I think the STORM study could only happen because of our long-standing relationship with Professor Altice and colleagues from Yale, that we were able to come together and design this study very quickly and now execute it. And hopefully we'll be able to add to the knowledge of how to manage patients at high risk of disease progression with COVID-19. So with that, I thank you very much, everyone, for joining us this morning in this forum.
Madam Ambassador, thank you so much for inviting me to participate. Adeeba, it's always great to share a podium with you and talk about this really important public health crisis. One of the things that has been really quite evident as part of this process is sort of all of the collaborations that we have that are really quite global. And in this case, it is really symbolic of that kind of collaboration that expands beyond what you think is your original mission to sort of move on to sort of new and emerging infections. Adeeba gave a really wonderful overview of where we are with regard to sort of the epidemiology and the treatments that are available. I'm going to transition a little bit onto COVID-19 vaccines, because I think that there is just a really unprecedented amount of work that has happened to sort of move this vaccine really to clinical trials and early efficacy studies that has never been seen before. So this really represents a huge, momentous sort of change, a real game changer. The fastest vaccine that we ever had that went through the process was Ebola, considerably longer. And when we presented just 6 months ago, we sort of said that we were online by the end of the year to have potentially some early results. And indeed, here we are. One of the things about the COVID vaccine updates is that, there is a COVID-19 tracker. And for those of you who are sort of interested in updates along the way, here's a website for you to go to. But just to sort of let you know about the magnitude. When we last spoke, I said that there were about 113 vaccines that were in development. We're now up to 236, and 40 of them are in clinical trials testing. And so this really represents a real game changer. And, I've heard people talk about who's going to get to the finish line first. And I don't really think that that matters, because the differences in the vaccines and the sheer magnitude of the vaccines are going to really, really be needed in order to come up with multiple options for maybe perhaps different patients. Now, one of the things that has been sort of at the center of this really big effort. Project Warp Speed is a US initiative. There are some global ones as well, but this is an effort that was referred to by Madam Ambassador to really sort of get at the issue of how to move this along in a really incredibly fast way. And what has been sort of really challenging with this is we've got on one hand the sort of the urgent need in order to sort of get a vaccine that is available. So we've got this speed phenomena, but we also need to make sure that we are sort of meeting the safety criteria. And for those of you who've been watching the news, we've seen a couple of these vaccine trials stop because of concerns around safety. Some of them have been assessed and they're back online. But those are always the issues. And for a situation like this, we really have to have something that is somewhat efficacious. And so we're really in an interesting sort of space with regard to this. Now, there are a number of major methods that are, some of them are completely new, but I don't really want to go into the details around the science of this and dig into the weeds too much. But essentially, the sort of classic design that we've thought about is we take a protein. So if we have an infection, we take that protein to it, we may break it off, or and then we sort of inject it in and the body makes antibodies to it, which is your immune system response to any kind of infection. But what's happened with these sorts of new sorts of innovations is that, you know, this spike protein, these parts of the crown, the thorny crown, if you will, those are the parts that seem to be the most immunogenic. And that's what you really need to make something to which you can eventually have an antibody. And so what they are doing is in some of these vaccines that are using a vector, they're taking the spike protein gene, the gene that actually codes for that. They're sort of amplifying it. They're in many cases modifying it, and they're putting it into other viruses. Adenovirus is one of the most common ones, but there are others. And then what ends up happening is these viruses are not pathogenic, or they are sort of mutated in a certain way, depending upon which of the vaccines. And it makes it so that the body expresses these proteins so that it looks like, for all the world, it's the coronavirus, and the body makes antibodies, but you don't actually get the sickness. And one of the things that I think is probably the most incredible change, and it's the two new vaccines that we've heard about in the press, and that those are these mRNA viruses, where they take these mRNA parts of the virus. They actually put them in a sort of a fatty sort of covering. They inject them, they fuse within the cells, they go into the cytoplasm, and then they are sort of altering the mechanisms within the cells so that they're actually creating proteins that have cells that have this particular spike protein on the outside of it, and then the body makes a system. So you're not putting any sort of live virus at all into the person. So here's a sort of a schematic for this. Here's the spike protein. We've got the coding for that, which is in the mRNA. They put it into a fatty cell or a lipid nanoparticle. They sort of inject it in, it fuses with the cell, and then it alters the protein recognition so that there is this spike that comes on the top of the cells, and then the body has an immune system to it, and it makes antibodies, and it promotes T cell recognition and response so that you can perhaps have a long-term response. Now, we've got two vaccines just all in the last week that have some really important findings. And, you know, I want to just walk you through a little bit about what we know, because it has major implications for how we're going to roll them out. And we're going to need them both. So there's no question about it. So, you know, a week or so ago, we had the Pfizer-BioNTech group that came up with the vaccine. What's really interesting is we've got Pfizer, which is the largest pharmaceutical company in the world. They did this without any government support. And they joined with this company in Germany called BioNTech. And what's really interesting is BioNTech has got the technology to put this messenger RNA into this this fatty nanoparticle, which is a technological advance in order to sort of get it into the body. Moderna doesn't use that sort of technology, but one of the things that's going to be really important as I get to the issues around the equitable rolling out of this virus, because it's one thing to sort of get through the science, and the efficacy's there. We're essentially pretty much there with regard to that. They're already manufacturing at risk hundreds of millions of courses of vaccine. The real issue is going to be the implementation. How do we get it to the right people at the right time in a timely manner? And so just with regard to these RNA viruses, one of the things about them, unlike these adenoviruses that are going to be the sort of the next generation, is that it has to be stored at freezing temperatures. And there are some differences with regard to the Pfizer-BioNTech and the Moderna vaccines. And one of them is that the Moderna vaccine can sort of be frozen in your freezer. Doesn't take any kind of special control. Now, the Pfizer vaccine, if you want it to last for 6 months, it's got to be stored at -70. There are very few of those, except for in scientific laboratories and distribution stations, mostly in sort of high income countries. And then if you take it down to the sort of standard temperature of your freezer, it lasts for only about 5 days. So you're going to have a cold storage chain system that will take them from the centralized units out to the periphery so that it gets to the people with regard to the Pfizer vaccine. The schedule for these vaccines is also a little bit different. So thinking about how you might implement this in communities. So the Pfizer vaccine is you get a first inoculation. Both of these require 2 inoculations. So, once again, you've got to sort of overcome the logistics of getting a second vaccine into patients. And what you do is you revaccinate 3 weeks later. With the Moderna vaccine, you vaccinate and then you wait 4 weeks later with regard to, 4 weeks later before you vaccinate again. Now, what's really interesting is, and I'll talk about the findings of the studies, but the way that they did these studies is they really only were able to detect patients. The difference was based upon the difference of patients who developed symptomatic disease. And where that is a potential problem is we won't know whether these vaccines are effective at reducing infections. All right. So if all of a sudden we think that we can sort of reduce patients from getting sick, that is, it modulates the way that the presentation of the disease is, versus actually shutting off the infection, it will have a different implication. So if we still have people who get infected at a similar rate, but they're not symptomatic, it means that we may not be able to shut off the secondary transmission. The data for the 2 vaccines is pretty superimposable with regard to its efficacy. The early report by Pfizer showed that it was 90% effective. They relooked at their data. The number went from 94 to 170 cases of COVID, and they were able to go back and rerun that, and it turned out to be about 95% effective. Very similar number, identical for the Moderna vaccine. The sample sizes were a little bit different. I would say that the stratification for the Moderna vaccine, better reporting. They gave a more comprehensive report. And remember, none of this is in the peer-reviewed literature. But in the Moderna vaccine, they had 11,000 of the 30,000 patients vaccinated or that were in the study. 50% of them were placebo, but 11,000 of them were people of color. 7,000 of them were in the high-risk category of being 65 years or older, and 5,000 were less than 65, but they had at least one of the standardized comorbidities that is associated with more severe disease. And what you sort of get at is a couple of really important findings, is that what they did was they waited until they got to a certain number of cases and they said, all right, let's unblind the study. Let's take a look at this and see which ones of the ones that are these symptomatic cases. And they confirmed them with testing to make sure that they were symptomatic with the right infection. And what they found is that, at least in the Moderna vaccine, that 90 of the 95 cases were in the placebo arm, and only 5% were in the vaccine arm. And what's really interesting that has a sort of a secondary effect on this is it seems to have a modulating effect, that there, at least among the 90 cases, there were 11 of them that had more severe disease. So that's exactly what you would expect. They would go on to have more severe disease, especially with this kind of profile. And the other sort of thing that you see here is that there were none, suggesting that even if you did break through and get an infection, you were going to have a less severe case. That kind of additional information has not yet been reported for Pfizer, so that's unknown at this time point. What I will say about both of these vaccines is that there were really no major unexpected side effects. It was the usual sorts of things around discomfort, maybe a little bit of malaise, a little bit of a headache, but no signal that suggested that there was any kind of severe side effects in these 30 to 40,000 patients in each study. What is important to remember is, once again, it does not, the study as it is right now does not demonstrate that there is a reduction in new infections, but at least with regard to symptomatic infections. And so when we get to a little bit more detail in the studies, we're going to be able to see the extent to which we're going to be able to shut down onward infection to others. Now, further down the road and not quite as far along in the clinical studies are these viral vector vaccines through a couple of different mechanisms. And what they do is, in this particular case, there is this gene within the coronavirus that codes for the spike protein. And what's really happening with that is that they take this particular protein, they either put it in a virus that doesn't really have any sort of effect, and they inoculate and the body makes antibodies to that, or they put it in and it's a virus that is completely non-pathogenic. It could be a human, let's say adenovirus, or it could be one that affects livestock. And then these virus gets into the body, it sort of changes the replication within the body so that this spike protein is once again exhibited on the replicating virus, and you develop an immune response. So the messenger RNAs have never been done. We do have a number of examples of the viral vaccines through adenovirus and other sorts of viruses, such as the Ebola virus and some of the actual HIV viruses that have been modulated. These are probably some of the leading ones that are working with adenovirus vectors, the one by Johnson & Johnson combining with Janssen and AstraZeneca and Oxford University. What's really interesting is you are actually putting in virus, but it's not a pathogenic virus. So people are not actually getting sick, and they're often using sort of non-pathogenic human viruses, which are the common ones, or ones from other animals that don't really sort of impact humans. And then, of course, the one that's being developed by Merck Sharp and Dohme and the International AIDS Vaccine Initiative does use a live vaccine, but it's not pathogenic. And it comes from a livestock sort of virus called vesicular stomatitis virus. The reason I bring up some of this is that people say, oh, you're putting in a virus and stuff like that. There's a lot of the safety checks that are being done because they're sort of turning on the machinery for replication so that spike protein is getting shown, but it's in viruses that don't seem to have any impact on humans. So now that we've got a virus that is being sort of developed, it's not quite approved, but we're pretty darn close, is that we've got to figure out what is going to be the proper allocation of these vaccines. And we sort of need to think about what is the impact of COVID-19 on the lives of people. And one of the things that we have to really start with and recognize is that there has been incredible inequity that has been the hallmark of the COVID-19 pandemic, both locally and globally, with really extraordinary suffering, death, and risks of death, and societal dysfunction, our economy, etc. And certainly in the US, some of those drivers have been some of the sort of the underscoring of racism and poverty, with our most disadvantaged communities being really at higher risk for having, one being at risk for getting the disease and then going on to developing more severe consequences and death. So we really need to sort of start out from that position of equity to make sure that the vaccine allocation must address those at greatest risk, as well as the drivers for more severe disease. And they must be supplemented by, other evidence-based practices, the things that Adeeba spoke about, really stopping the large congregations of individuals, using your sort of facial coverings, and all of these things. So this is not going, these vaccines are not going to be a silver bullet, because they're not going to be able to be put into the community at a fast enough rate to have a major impact. So all of those other sorts of things are really at the root of what we need to do until this is able to be expanded over a series of several phases. There will be planning that will need to happen at both international and national levels. And there will be major issues about deciding who can get the virus and what are the kinds of extraordinary amounts of money that are going in to make sure that the have-nots are able to get it through purchasing power, et cetera. It's estimated that the Pfizer vaccine is going to cost about $19.50. And in some communities, that is the overall expenditure for health per person for an entire year, and it may be as much as 3 or 4 people. So we also need to know that the planning that will happen more globally. The real work is going to happen within states, localities, and in smaller sorts of settings. So that will be where the rubber hits the road with regard to the way that we dispense this. So, one of the ways of thinking about the equity of this virus is looking at those who are at highest risk, not only for disease, but for the consequences. And certainly we know that the most severe consequences occur in those individuals over 80. Their risk of death is 5 times greater. If you drop that age down a little bit to 65 or older, if you're over 65, that accounts for 80% of deaths, but certainly not all of them. And remember, death should not be the only indicator. It's just the one that's permanent, right? And so in this group, the risk of death is 16 to 52 times greater in the US, and globally somewhere between 30 and 100 times higher. If you're in a high income setting where you've got lots of mechanical ventilators, you will have a lower mortality rate. And so, that's going to be a major issue. Having an underlying comorbidity, obesity, diabetes, our whole list of cast of comorbidities, that increases your risk for hospitalization 6 times and for death 12-fold, and that's in the US. If you happen to live or work in a congregate setting, that is related, especially like nursing homes, et cetera. That's an elevated risk for severe disease and death just by virtue of who is in those settings. And that accounts for about 50% of deaths in the US. And then certainly we know that people of color have an increased risk for getting the infection. They have an increased risk for hospitalization, and they have an elevated risk for death overall, looking at aggregate data. So what might be the elements of a fair allocation strategy? And that is, can be easily and understood by diverse audiences. So in other words, it's got to be understandable. If people are afraid that they're getting a vaccine that has maybe perhaps something that came from a virus, will they accept it? It needs to be sort of accepted under social and ethical principles. It needs to be reliably translated into operational terms. What does it mean if you get this? What are your risks, et cetera? And there have got to be a number of factors that does not perpetuate discrimination and further inequities through that allocation. The overall goal is to sort of reduce new cases, but also severity of diseases and the consequences. There's got to be some allocation criteria that's based on the sort of things that we talked about. And then the other part of this is going to be the allocation that's based on what I call social harms. That is, people not being able to work or shutting down certain parts of the economy and those sorts of things, because that is you have to balance the societal and the human capital risk with regard to this. Part of the sort of allocation process is sort of focusing on the factors that are associated mostly with death as an outcome that is irreversible. So when balancing some of these things, I have 7-year-old twins, and so there is a societal impact to school children and non-essential workers. There may be a downturn in the economy, and some of these effects can be partially reversed, not permanently, but partially, whereas death is not that way. And we certainly need to prevent some of the disease severity, and we need to sort of have really reasonable data to suggest that that we can look at other groups of individuals who are at risk, such as healthcare workers in congregate settings and other sorts of places. Here is what might be a sort of a phased-in approach to vaccine allocation, and this has been the one that has been developed for the U.S., but there's a jumpstart phase. We know that we're going to get a very limited amount of vaccine, and the phase 1 will cover about 10% of the people that you need to cover. And it will be really focusing on high-risk healthcare workers, that is people who were right at the scene, the first responders who go pick up people out in the field. Phase 1B, which is still part of phase 1, is patients with underlying comorbidities and significantly higher risk for disease. And then, of course, older adults in nursing homes, et cetera. With the second phase moving to teachers and school staff, critical workers who are in socially vulnerable situations, et cetera. People who are in closed settings like prisons, jails, and detention centers, and then some of the older individuals who are not there. Then moving into phase 3, which is getting most of the rest of the people. And then phase 4 is circling back and saying, who have we missed over time? I'm going to sort of move out of this, but, what has to happen is that places have to adopt this. We learned this with HIV. When we had a limited amount of antiretroviral therapy, we have to come up with sort of coalitions to sort of prioritize who gets medications first, and it has to be really sort of prioritized, and it needs to be transparent. Ideally, we would like to make sure that when we're doing this, that we have a method for distribution and administration. There is no time like the present to have all of those things put in place, such as our cold storage chains, making sure that we are purchasing it and we're not only giving it to the people who can pay for it, but that it's allocated in an equitable way, so avoiding any out-of-pocket costs that we possibly can. We need to make sure that we've got a COVID-19 vaccine promotion campaign. There will be lots of skeptics, and if we have a high level of skepticism, which we already have in the US around vaccines, or we don't have systems to get vaccines to people. I know in my own community, we have launched an influenza vaccine in our 130,000 person community. 85,000 people did not get an influenza vaccine. So one, they have to think it's important, they need to think it's accessible, and they need to think that it's going to be able to be safe for them. I will end on the note from a Nobel Prize winner in physics who did a lot of really important work in quantum physics, Richard Feynman. Certainty is the Achilles heel of science, and if I get to talk to you in 6 months, some of these things that seem almost certain today may not be certain tomorrow. Thank you very much.
Well, Prof, I'm sure every one of us hope that when we speak to you back in 6 months, things are getting much, much more better.
How do I turn off my screen? Or not share my screen?
Thank you so much, Prof Altice and Prof Adeeba, for enlightening us on this topic. We shall go to the comment thread on Facebook and find out what the members of our audience want to know more about. The first question, and this is a question that I'm quite interested in as well, is that when will we know how effective and for how long the protection a vaccine can provide?
Rick, you're muted.
Muted.
All right, thank you so much. This is a really, really important question. And so, part of the ways that they've developed these antibodies is so that you would actually have not only a direct response to the pathogen, but you also have a response by T cells, et cetera. And, we don't even know for sure that even getting the infection will result in long-term immunity. We certainly know that you seem to be protected for, at least 90 days. This may be the situation like influenza, where you vaccinate people 1 year and you're going to have to repeat it again later on. But we will not know that from the early phases. We will know how to have at least a short-term benefit. And we will not even know, at least immediately, whether we're going to be able to blunt new infections. We will only know whether we only know now that we can blunt symptomatic infections. And so that's a really important question. So we really are going to have to follow all of these people over time. And so that's part of this vaccine trial. So this is the early efficacy, but we're going to have to look at the holy grail is, does it actually prevent infections, and is it sustainable? Very important question. We don't know that yet.
All right, thank you, Prof. The second question we have here is that when can we realistically expect the COVID-19 vaccination program to begin in Malaysia?
So I don't know how the allocation has been decided for Malaysia. I know that the European Union has already planned for several tens of millions of vaccine. The US has got 100 million vaccines. Only 50 million of it will be available. You know, they're going to, Pfizer has already committed to, by next year, being able to put out 1.2 billion vaccines. You know, it's the largest pharmaceutical company in the world, so their ability to manufacture is going to be probably better than Moderna. Moderna's got a very, very fine vaccine, and the amounts that they're going to be able to produce will be later. So it all is going to be dependent upon the rate and then the country buy-in. How much is the country ready to spend, and how much is the international donor community willing to do that? And Malaysia, as an upper middle income country, is going to be expected to come up with probably more money than, let's say, a lower middle income country in order to sort of come up with that. And if we're talking at $19.50 per vaccine, that's not an insignificant amount of money.
So if I can add to that, as far as I know, Malaysia is a signatory to the COVAX consortium, which is a consortium of, led by WHO and CEPI and Gavi as part of the overall international movement that includes the World Bank and other large donors in what's called ACT Accelerator. So the ACT Accelerator aims to, make tools, diagnostics, vaccine treatment as quickly accessible to the haves and have-nots parts of the world. So this COVAX is the vaccine arm of ACT Accelerator. But as Rick alluded to, Malaysia, that within the COVAX itself, the low-income countries will be supported in terms of financial support towards access to the vaccine, but Malaysia will, I believe, have to pay for it. So it depends on how much the government has committed to it, and also how far back we are in the queue for the vaccine.
Right. Thank you, professors. There's a question over here that, because when we speak about the Pfizer vaccine and we speak about the cold chain logistics that's going to be required to transport this vaccine to the population, there is a question by one of our viewers here.
Sean, I didn't hear any of that.
Right, sorry.
You froze, so you need to repeat the question, please, Sean.
So we spoke about the Pfizer vaccine just now, and we spoke about the cold chain logistics that's required to transport these vaccines to the people. There's a question by our audience here who asks, what happens if a healthy person is injected with a Pfizer vaccine that has been unknowingly not stored under negative 70 degrees Celsius? Would we know that?
Well, I mean, that's going to be sort of responsible for what I call the cold chain storage, about sort of only giving vaccines that are going to be, sort of safe. So if you want to think about what that would look like, is the people who are monitoring that. I mean, will it hurt you? The answer is no. Will it probably have any sort of benefit? It depends on how long it's been out. But, you've got 6 months at minus 70, so you can put them at these centralized repositories. You got 5 days at putting them in your sort of freezer in your kitchen. And then, you'll only get 5 days out of that. Once you go beyond that, we don't know that the vaccine will hold up, much, much longer. So you probably won't get harmed by it, but you certainly may not get the benefit. So you might be falsely allured to thinking that you're getting something.
Right. All right. Thank you for answering that question. There's another question over here. Is it possible that sometime in the future, the treatment of COVID-19 can take place in an outpatient setting?
Yes, I think that's a very good question. Certainly, the dexamethasone is an oral pill, which is widely available, and it's inexpensive. And but of course, it like all medications, it has its side effects. But I think The key would also be in terms of improving testing. At the moment, the PCR test is not easily available and accessible in primary care settings, which makes, someone who suspects themselves as having COVID-19 kind of not so can walk into any primary care and say, may I have the test? So I think there are a few things that need to happen in order for treatment with dexamethasone to be available in primary care setting, and that's point of care tests for the disease, the virus and the disease. And this is not so difficult, a clear sort of treatment guidelines based on risk assessment and the potential for disease progression, and sort of have a criteria for disease. That can already happen, but I think the rate limiting factor at the moment is the test.
And I would just add to that and say that we certainly need an oral antiviral, just like we have for influenza, sort of oseltamivir sort of thing, where you take it. So we need to be able to have a rapid test and treatment early on, but we're still already at the stage of late disease, and in the corticosteroids work when patients are starting to get to the point of needing intubation. So it's going to be a fine line. So for us to be able to predict when somebody's going to be heading in that direction is going to be sort of a key. But you're absolutely right. We have to have the testing, and we have to have the ability to sort of identify the people who are progressing. Right now I'm using for my outpatients, because I'm trying to keep them out of the hospital, I have them walk and get a pulse oximeter, but that's not going to be possible for most of the world. You can't even buy them on the shelves anymore in the US. And so we're going to need to have those tools in ambulatory settings to do that.
True. For now, we need to test, and test. That's extremely important. I think we have time for one last question, and this is a long question. As the COVID-19 pandemic has been going on for nearly a year, many people may have already been exposed to the virus by now, be it being symptomatic or non-symptomatic. Will the vaccine still be able to protect these pre-exposed individuals from severe symptomatic infections in the future?
So let me just say something about the people who might have been exposed, let's say back in February or March. We don't know that they're still protected today. So let's be very clear that we don't know that yet. And so the question is those are people that I still think should really be, considered for vaccination. I would not stop that based upon, their sort of prior exposure, because we do know that there are cases that break through. And at least if you look at the Moderna vaccine, where they broke down, the findings there seems to be the marked reduction in the severity of disease. So it may have an immunomodulating effect in addition to a protective effect. And so I think that we're going to have to, all we've got are sort of reports that were given to the press. We have not been able to dig into the details of these studies yet. So, what I'm presenting is based upon these sort of reports that the companies release, not even by peer review. But it's promising. It really is promising. I think that's really the bottom line.
All right. So thank you, Prof. Thank you, Prof Adeeba. Thank you, Prof Rick. While we're having a great time here today, I'm afraid time is our greatest enemy. We have now come to the end of our session today. Once again, thank you, Prof Adeeba. Thank you, Prof Altice, for your time. I shall close our session today with a Malay poem, otherwise known as a pantun. Langit megah berbunga api, dipawali disambut berwarna-warni. Rakyat bersama ikut SOP, dengan ukhuwah COVID pasti ditangani. Wear a mask, stay physically distant, sanitize your hands. Thank you everyone, including our friends from the media, for gracing us with your presence. Have a good day all.
Thanks very much, Sean. That was lovely.
Thank you.
Thanks everybody. Good job.